Welcome back to the deep dive. Today, we're tackling something incredibly intricate, the human brain, and specifically how we're starting to map it out in autism spectrum disorder, or ASD.
It's a huge area and really important too.
Definitely. Especially when you hear about the rise in ASD prevalence in kids over the last 20 years or so. I mean, the numbers jumped from about 6.7 per thousand to nearly 28 per thousand in the US.
Yeah, that's a significant increase.
It really underscores why we need to understand this, the neurobiology behind it, what's actually going on in the brain. But the big challenge for researchers has always been just how complex ASD is.
Right.
It's a neurodevelopmental condition, sure, with differences in social communication, restrictive interests, repetitive behaviors. But the key word is heterogeneous.
Mm-hmm. Hugely varied.
Exactly. How it presents is so different from one person to the next. And even with all the amazing neuroimaging tech we have now, well, the findings from brain scans have often been really mixed.
Yeah, inconsistent is a good word for it.
It's like different studies keep finding different things.
And that's precisely the puzzle we're digging into today. We're drawing insights from this really comprehensive review by Halliday and colleagues just out this year. They looked at a ton of research, 172 papers from 2013 to 2023.
Wow, that's a lot of data.
It is. And they covered 7 different ways of looking at the brain. You've got structural MRI, functional MRI, DTI,
Okay.
MRS, FNIRS, MEG, and EEG. Basically 7 different lenses. And they use these to compare the brains of autistic young people under 18 with typically developing kids.
Trying to find some common ground, presumably.
Exactly. They were looking for both heterogeneity, where things differed, and convergence, where, surprisingly, different methods might actually point to the same thing.
Okay, let's unpack this then. I know some of those names sound pretty technical.
Yeah, a bit of alphabet soup.
Yeah. But we'll break it down. We want to get to the core insights so you can feel really informed. So, to kick us off, why has it been so hard to find consistent patterns? With 172 studies, you'd think something clear would emerge. What did the reviews say about these mixed messages?
That's really the starting point. The review emphasizes this considerable heterogeneity. Lots of variation across all the different imaging types. And what's really striking is that differences between the autistic and typically developing groups, they were rarely defined in one direction.
Meaning?
Meaning you wouldn't consistently find, say, one brain area always being bigger or always smaller in the autistic kids. It varied study by study, sometimes bigger, sometimes smaller, sometimes no difference.
Huh. Okay, so it's not a simple A versus B difference. What did the paper suggest were the reasons for these inconsistencies? Were there specific hurdles?
Yeah, they pinpointed a few key factors. First, just huge variability in the study populations.
Like the participants themselves.
Exactly. Different studies looked at different age groups or had really wide age ranges. And a big one, many studies didn't consistently report or match participants based on sex or gender or IQ.
Right, and those factors definitely influence brain development.
Hugely. So if your groups aren't comparable on those measures, you're bound to get different results. Second factor, evolving methods. Neuroimaging tech has changed so fast.
Sure.
We've moved from just looking at one specific region of interest to doing whole brain scans. And the scanners themselves are better. Which is good, obviously, but it adds, well, another layer of variability when comparing old and new studies.
Makes sense, like comparing photos from different cameras over decades.
Precisely. And the third thing was processing differences.
How they analyze the images.
Just a really broad range of choices researchers make. How long the scan was, how they dealt with movement artifacts, even the software they used. All these little decisions can nudge the results one way or another.
Okay, so different participants, different cameras, different photo development. No wonder the pictures look different.
Right. But what's really interesting, and you touched on this, is that this heterogeneity isn't just a problem. The review suggests it might actually reflect real, true biological differences among autistic young people. It fits with the whole idea of neurodiversity.
So the diversity in findings reflects the diversity in people.
Exactly. Maybe we shouldn't expect one single autism brain profile. The diversity in the brain might mirror the diversity we see in behavior and experience.
That's a really important perspective shift. Okay, so given all that variation, let's dive into what each specific technique did show. What were some of the insights, even if mixed? Let's start with structural MRI, fMRI, that looks at the physical structure, right? Gray matter, white matter, volume. What did those 26 papers generally find?
So one of the more consistent findings from fMRI was actually a larger overall subcortical gray matter volume in the ASD groups.
Subcortical, so the deeper brain structure.
Yeah, areas involved in emotion, memory, basic drives. That was interesting because a lot of early focus was on the cortex, the outer layer. But for the cortex itself, findings were much more mixed. Some regions showed larger volume, others smaller. So again, that heterogeneity pops up.
Okay, so deeper structures potentially larger, but the surface is more varied. What about diffusion tensor imaging DTI? That's the brain's wiring, the white matter connections. 14 papers on that.
Right, DTI looks at white matter integrity. And the main trend there across studies was overall higher mean and radial diffusivity in the ASD groups.
Which suggests.
It suggests lower white matter microstructural integrity. Think of it like the insulation on electrical wires being less consistent, or maybe the fibers not being as tightly packed. It could mean less efficient communication between brain regions.
Got it. So maybe the connections are there, but they're not as smooth.
That's a decent analogy, yeah. Now, functional MRI, fMRI had the most papers, 41. This one looks at brain activity and how regions connect functionally. What came out of that?
Yeah, fMRI results were described as having substantial variability, highly mixed.
hmm But some patterns did emerge, especially in the larger studies. They found higher connectivity within certain subcortical regions. Those deep areas were busy talking to themselves, maybe. But lower connectivity in networks like the default mode network, the DMN.
The DMN, that's the network active when our mind wanders, right? Self-reflection, thinking about others.
Exactly. So less connectivity there, and also less in the ventral attention network.
Yeah.
But then lots of findings about connectivity between different networks. Key hubs like the amygdala, thalamus, insula, prefrontal cortex often showed higher connectivity to other areas in ASD groups.
Higher, so more crosstalk.
Often, yes.
Big
So not
difference.
always. Sometimes
Yeah.
it was decreased, like between the amygdala and the visual cortex. So it suggests an atypical pattern of how different brain systems are coordinating or maybe not coordinating.
Fascinating. Okay, moving to brain chemistry with magnetic resonance spectroscopy, MRS 19 papers. What metabolites stood out?
MRS gave some really interesting clues. A key finding was evidence for higher markers of excitatory neurotransmission, think glutamate, the brain's main go signal.
The accelerator.
Right. And lower markers of inhibitory neurotransmission like GABA, the main stop signal.
Brakes.
Exactly. So this points towards an imbalance. Maybe the brain's signaling is tilted towards excitation. This fits really well with the excitation inhibition theory of ASD.
That makes sense. Anything else from MRS?
They also found some signs of lower markers of overall brain health and metabolism, suggesting potential differences in how brain cells are functioning or getting energy.
Okay. Next up, functional near-infrared spectroscopy, FNIRS. 20 papers. This tracks blood oxygen levels.
Yeah, it's a proxy for neural activity. More oxygen use means more activity. And the overall finding here was lower oxygenated hemoglobin levels in ASD groups, especially in areas like the prefrontal cortex and temporal regions during tasks.
So less activation when doing certain things.
It suggests decreased task-related activation, particularly for tasks involving social skills or executive functions, like those brain areas weren't ramping up as much.
Interesting. And finally, let's group MEG, 20 papers, and EEG, 32 papers. Both measure electrical brain activity, brain waves. What did they show together?
A really consistent finding across both was decreased auditory event-related potential amplitude.
ERPS. Those are the brain's automatic electrical response to a stimulus.
Yes. So when presented with a sound, the typical electrical spike was smaller or less pronounced in the artistic groups. For EEG, this was also seen for visual stimuli.
What might that mean?
It could relate to how sensory information is initially processed, maybe tying into sensory sensitivities or differences. The brain's immediate reaction seems altered. Other findings were more mixed. EEG showed some evidence for higher overall connectivity across the whole brain, but also generally decreased frequency or power, slower brain waves in many areas.
Okay, wow, that's a whirlwind tour of 7 different techniques.
It is.
But here's the really exciting part you mentioned earlier. Despite all that variation within each method, the review found that certain brain regions just kept popping up again and again across different methods, these hot spots.
Exactly. This is where it gets really compelling. Despite the noise, despite the heterogeneity, these specific brain areas consistently show differences, regardless of whether you were looking at structure, function, chemistry, or electrical activity.
Okay, let's list them out. Which areas were these consistent players? First up, the anterior cingulate cortex, the ACC.
Right, the ACC, involved in decision-making, error detection, emotion. It showed higher markers of excitation, like we saw with MRS, and also altered functional connectivity patterns from fMRI.
Some multiple signals pointing to differences there. What about the insula?
The insula are critical for integrating bodily feelings with emotions. It showed altered functional connectivity. And interestingly, the right insula specifically showed both increased white matter volume and decreased cortical thickness. A complex picture.
Definitely complex. Then the prefrontal cortex, the PFC, our executive control center.
The PFC showed up repeatedly. Altered resting state connectivity in fMRI, those lower oxygen signals during social tasks in FNRS, and some structural studies found less gray matter in certain parts.
Makes sense given its role in social cognition and planning. Now, the amygdala, this one seems really central in many discussions.
It really is. The amygdala consistently showed larger volume, which is a finding that goes back even to infancy studies. But paradoxically,
Yeah.
it also showed decreased functional connectivity with other regions important for emotion processing. So it's like this key emotional hub is larger, but maybe less well integrated into the wider emotional network.
Which is a paradox. Bigger but less connected. Okay, the thalamus, the sensory relay station.
The thalamus showed increased functional connectivity,
Yeah.
especially with sensory areas like the auditory and primary sensory cortex.
So maybe over-communicating sensory information?
Could be related to sensory processing differences, yeah. Like the relay station is amplifying or sending signals differently.
And the cerebellum. We used to think just movement, but it does much more.
Absolutely. The cerebellum showed decreased resting state functional connectivity and also that lower GABA ratio, pointing back to the excitation-inhibition balance. Plus, that really specific finding about lobule 3 changing volume with age smaller before 11 than larger suggests an atypical developmental path.
Very specific. What about the bridge between the two brain halves, the corpus callosum?
The CC had mixed findings, which maybe isn't surprising given it's a large structure. Some studies found larger volumes, others lower white matter integrity, some even higher rates of it being underdeveloped hypoplastic CC. So variability again, but consistently studied.
And lastly, the default mode network, the DMN, came up again.
Yes, the DMN consistently showed significantly altered connectivity with other brain networks. And some of the brain structures within the DMN showed larger volumes. It really reinforces the idea that the brain's baseline state, how it operates when inward focused, is different.
Okay. So, seeing these regions, ACC, insula, PFC, amygdala, thalamus, cerebellum, CC, DMN, pop up across different types of scans, that feels significant. How does this connect back to the bigger theories about ASD?
It connects really strongly. Take the excitation-inhibition imbalance theory. The MRS findings, the ACC findings, the cerebellar findings, they all add evidence supporting that idea.
Right, the accelerator brakes idea.
Exactly. And the widespread findings of altered connectivity, both higher and lower, within and between networks, strongly support the idea of atypical connectivity patterns. What this review suggests is that maybe it's these differences in connectivity, potentially at a microstructural level, how the wiring is actually set up, and functions that might be more central to the neurobiology of ASD than just changes in the size or shape of brain regions.
So it's less about the destination size and more about the road network connecting them.
That's a good way to think about it, yeah. How efficiently and typically information travel seems key.
So, bringing this all together, what's the big takeaway for you, our listener? What does all this complexity but also these converging patterns mean for understanding ASD?
Well, I think the main message is that the brain in ASD is complex and diverse. There isn't one single autism brain. Understanding that heterogeneity is crucial. But these converging patterns, they give us really valuable clues about the underlying neurobiology. They help us appreciate that rich neurodiversity we talked about. And practically speaking, it's vital to remember these findings aren't exclusive to ASD. Some overlap with anxiety, ADHD, sleep issues.
Right, things that often co-occur.
Exactly. So the review is very clear. It is inappropriate to assume that all autistic youth have similar neuroimaging phenotypes. And you definitely cannot diagnose ASD based on a brain scan alone. The brain findings are part of a much bigger picture.
That's a really important caution, but does this complexity, this heterogeneity, offer any hope for the future? Could it actually be useful?
Potentially, yes. That's one of the exciting implications. If we understand that brain profiles are diverse, maybe we can use that. For example, the review mentions leveraging this heterogeneity for personalized targeting in therapies like RTMS, repetitive transcranial magnetic stimulation.
So tailoring treatments based on an individual's specific brain patterns.
That's the hope. Well, this review didn't directly link specific brain differences to specific ASD symptoms. It does highlight potential targets that future research could explore for personalized interventions. And looking forward, the review calls for researchers to pool raw data for meta-analyses to get more precise estimates. That wasn't possible here. And crucially, there's a growing call to include ASD stakeholders, autistic individuals, and their families right from the start in designing research.
To make sure the questions being asked are actually relevant.
Exactly. Research with the community, not just on the community. That's becoming increasingly important.
Absolutely essential. Okay, so wrapping up. The brain in ASD, it's marked by a lot of variation, inconsistent findings across studies, but underlying that noise, there are these converging patterns across different ways of looking, different brain regions, different ages. It's not one simple picture, but maybe a complex, diverse mosaic.
Well said. And it leaves us with a really interesting thought, doesn't it? Given this huge diversity within autism, maybe the key isn't trying to find the single autism brain. Perhaps future research, especially using multiple imaging methods together, should actually embrace and study this heterogeneity. How could understanding that diversity unlock truly personalized insights and supports that actually work for individual autistic people?
That's a powerful question to ponder. We hope this deep dive into the complex world of neuroimaging and ASD is giving you some new perspectives. Thanks for joining us, and we'll see you next time for another deep dive.